My name is Ainemurwani, a student at Rwentanga Farm Institute, pursuing a national certificate in agricultural production, second year, that is from 2023-2024. I'm humbled to have been named one of the best innovator at Rwentanga Farm Institute under the Startup Garage program, under the project of Spore production unit that produces mushroom seeds. Today I'm going to take you through how to come up with the mushroom seeds or the spawns, and we are in the laboratory. I'm going to take you through. Mushroom seeds undertake 3 steps. That is the one for making the culture, the mushroom culture, and another one for taking the mother spawn, and that's the one for seed bed spawn. The culture, it's where we are growing the tissue of the mushroom. Like here we are having this mushroom. The mushroom, the spores, they are tiny, you cannot see them unless maybe we have used the microscope, but usually we opt for the tissue of the mushroom. The mushroom has 2 layers. So when we get a sterilized blade, we cut these tissues. We are having 2 tissues, the outside and the inner one. So we cut the inner one that contains the spawns, the spores, and we remove it. So specifically, this is where the culture starts from. And the culture, we will have prepared a PDA, that is the potato agar dextrose mixture, where we're having here the agars that are fully mixed, that will allow us boost the growth of the tissue, this one. So from there, we put it, when we have prepared our mixture, that is the growing medium of the PDA here, we put this tissue in the mixture, and from there, we cover it. So when we cover it, it will grow and give us something like this, where we will find that this tissue will have grown. We'll see the threads growing here. So this, after 14 days, it will have provided us with this one, and this is the mushroom culture. So this mushroom culture, you cannot plant it. We need to put it into the grain so that it provides us the mother spawn. So from here, we are going to take this tissue that we have prepared, that we have put into the growing medium. We are going to take it to the incubator, where it will take 14 days, and from there it will grow. It will have produced the mycelium. The mycelium will have grown. So from there, after we have prepared the growing medium and we have put the tissue into our agar plate, we shall bring it and we put it into the incubator, where it will incubate from. So we are going to put it here. By 14 days, it will be under the temperatures, the normal temperature, that is 35 to 37. Those are the human body temperatures. That's the range where the mycelium usually grows faster. Then from there, after the 14 days, it will have grown. It will have covered the growing medium like this one. So from here we will have obtained the mushroom culture. So this mushroom culture, we are going to put it into the grains, produce the mother spawn, and from there we are going to get the mother spawn and we produce the seed bed spawn, which will be for planting. So after we have put this one, it will take the 14 days, and it is ever on the electric power, so that it remains with the temperatures for the mycelium to grow faster. Then from there, now when the 14 days are done, we get our grains. So these are our grains. Specifically, we are using the millet grains because it is locally available, and it is in the second production. If we don't have this one, we use the barley, but now we are going to use the grains. So these grains, first of all, we need to soak them and clean them. So when we soak these ones, we sterilize them, we soak and drain. Then from there, we have cooked them, because this one, we understand that they are growing. So when we have cooked them, we will have destroyed the radicals and the primals that are supposed to be growing. So after that, we sterilize them using this machine, which is called an autoclave. So this autoclave So it sterilizes about 126 degrees for 30 minutes. So we bring our pre-cooked grains, we put them into these buckets, then we arrange them. This autoclave, first of all, it's sterilized by steam, so it uses water. It produces water, and the water that it heats produces steam. Then it sterilizes for about 30 minutes, and from 30 minutes, our grains are sterilized and ready to be inoculated. So from here, this is a sterilizing equipment that uses heat. We have a sterilizing equipment that uses the UV lights. That one provides us with a free pathogen environment where we inoculate from, where we transfer our culture that has grown, that has taken for 14 days into the grains. So we're having that as the safety cabin. So from sterilizing, we bring the grains into this machine. So it is called the safety cabin, and its role, it provides us a free pathogen environment. So it is having the two bulbs, it is having the UV light, and it is having the UV lights. So the UV lights are the ones that sterilize the environment where we are working from, and this glass prevents the entry of the pathogens from outside. So the grains are put here and they are allowed to cool in a safe environment, and from there we get the other culture, we transfer it, we put it into the grains to come up with the mother spawn. So we are using the inoculating group to transfer the culture from there, and we bring it into the heart, into the mother spawn. So with this inoculating group, when you put it there, it brings only the grains, it brings the mycelium alone, and you inoculate it into. So you inoculate it into the grains. So after doing so, the mother spawn will raise it into the glass bottles like this one, because it needs a lot of care. Then the mother spawn also, when we have inoculated the mother spawn, we also take it into our incubator for 14 days. So when we take it into the incubator for 14 days, it will also have covered, the mycelium will have covered all the grains. From there, we repeat the process of the grains. We prepare the grains, we sterilize them, we bring them into this machine, where we get now the mother spawn, we inoculate it into the grains that we have prepared. Those grains will lead us into the seedbed spawns. So the seedbed spawns also, the process is the same as the mother spawn, but for it, we are relying on the mother spawn, not on the culture. Then from there, after we have inoculated from the mother spawn into the newly prepared grains, we again put them into the incubator for 14 days. So from there, remember, we have prepared the grains for the second round. Remember, the first grains gave us the mother spawn. Now that we have prepared the grains for the second time, they will give after 14 days. Now for the seedbed spawn, we pack it into the polythene bags, because this one will ease the farmers outside there to remove the polythene bag and get the seeds inside. The seeds, it is the white thing that you are seeing, the mycelium that we extract from the other tissue that we started with when we are making the culture. So after 14 days, we have got a product like this one from the mother spawn. You will find that a total of around 42 days are required to have this, because every process takes 14 days. And after those days, we'll have the seedbed spawn. It will be this one that we shall be planting, where under the mushroom production. So this was mushroom growing, but specifically level 3 of seed production. So after those days, we shall have gained these seeds from our incubator. They will have grown and ready for planting. So I'm happy, and I thank I myself for being committed to this project. Within the Startup Garage program, I was able to be the best innovator at Rwentanga Farm Institute with the Yuga Spore production unit, whereby the money that I got, I used it to improve on the quantity and the quality of the mushroom seeds. From there, I thank the Rwentanga Farm Institute team for having provided all these machines that I'm using. Basically, before I was not using these machines, I was using local means. I was improvising with local means, whereby where I needed like a sterilizer, I was using a saucepan. From there, I cannot forget to thank the 8 team for having programmed the startup garage, because it has brought me somewhere. It gave me the capital that I invested in. It also gave me the platform where I explained, where I had the customers from. So I thank the 8 team for having programmed and thought about the youth. So I encourage the youth also be creative, be innovative, and have the passion and commitment to the projects. It's the hands-on that will take us somewhere. Thank you.